Thank you so much. It's great to be here with you today. And thank you, Dr. Cowart, for giving that great introduction to lead into what I'm going to talk about today and about why we love this wonderful product that we call peanut so much. If you had trouble understanding Darlene's accent, you'll have trouble with mine too. I'm also from Georgia and southern U.S. I'm representing the Peanut Foundation. The Peanut Foundation is affiliated with the American Peanut Council, and we exist to fund research to make this product we call peanut, which is a wonderful product already. Our goal is to make it even better, to make this great product even better and have traits that we all can agree that will improve the product. That's why research is so critical. We all have competition. You business people here, you know that peanut is a great product, but we have competition from other products as well. We have production issues sometimes that hamper the flow of peanuts. So we're interested in funding research to help alleviate some of those problems and make this product even better. The first thing we need to understand, of course, is that genes control everything. This is not true just with peanuts, but with every organism. And science has advanced to the point that we know a lot more than we used to about genetics and about how genes control how an organism looks, how it functions. Peanut is no exception, and we know that all of these traits listed here and many, many more are controlled by genetics. Not all peanuts are the same. We know that. We know the different market types of peanuts are different. We know that even within a market type, the varieties of peanut that we grow have different traits. So we all know that this is due to genetic variation. The Peanut Genome Initiative was started about 6 years ago. The U.S. peanut industry got together and decided that they wanted to invest in understanding more about the peanut genome, the collection of all the genetic material of peanut. So the industry made a commitment, committed $6 million to the product. An alliance of scientists was put together, and this was a very international alliance of scientists that began to work together to try to accomplish this goal. I can say now that 5-year initiative is completed. We have a very good map of the peanut's genetic material now. The goal was to develop tools for breeders so that breeders could have a better insight into how to better put traits into their new varieties, and to develop markers. Markers are simply places on that genome that we can quickly test for and that are associated with those traits, either with a gene or with a group of genes that are associated with a specific trait. And we can do a quick genetic test, know that we've got that marker, that we have that trait. So that was our goals, and we're very pleased to announce that 5-year project is completed and has been very, very successful. So what is a genome anyway? Like I said, it's just the collection of all the genetic material in an organism. It's a complete set of DNA. And a lot of people immediately think, when they hear that word genome, they think, whoa, you're talking about genetic engineering, you're talking about GMO products, and my customers are not going to want a GMO. So we're very aware of that fact, and the industry has consciously made the decision over the last few years, even though a lot of other crops have gone the way of genetic modification, which basically means taking genes from one organism and putting them into another organism and incorporating those new genes into the genome. We have consciously avoided that technology. So we're going at it from a different way. So even though I'm talking about working with a genome and working with genetic material, it's important to understand that this is not technology that leads to genetically modified organisms. So we know that our customers, the ultimate consumer out there of peanut, is very interested now in more natural products, right? Everybody wants something natural. So here's this beautiful scene of nature, and the fact is that most of us don't eat all natural foods, even if we try. The only natural foods out there, if you're a hunter-gatherer, if you're a hunter and a fisherman and collect berries out of the woods, then you're eating natural foods. But otherwise, most everything we eat has been modified over time in some way. So look at that organism right there. You ever taken a walk in the woods and found those wild beasts there? Those are an example of domestication, of animals that have been changed over time by selection. Humans have selected traits in those dogs that they desired, and over time, thousands of years, we have all different breeds of dogs out there that they're not genetically modified, they're not GMOs, but they have been modified over time by human selection. So this is a natural peanut. Okay, these are wild peanuts that exist in nature, and there's many different species of them. They've been around for a long, long time, and they still exist. And I think you could look at those and say that you probably would have a hard time selling that product to your customers. They're not particularly desirable from the standpoint of a human food. But over time, humans have taken those initial wild species. We have selected traits that we are looking for, and we have modern day peanut. So there is a big difference between domestication of a product and GMO technology. Modern day peanuts are clearly delicious, nutritious, wholesome. Peanut Genome Initiative research led to an article in Nature Genetics that showed that we now know how those wild species actually naturally combined, two of those species naturally combined about 10,000 years ago, and modern day peanut began. And that process is well understood now. We know that these wild species that still exist in nature have a lot of beneficial genes. Even though that final product of a wild species may not be a very desirable peanut, they have desirable traits like disease resistance in those wild peanuts that we can use in modern day peanut to make our modern day peanut even better. So what is the advantage of the research that we're doing, the marker-assisted selection research? All of the plant genes are contained in the genomic map, but now that we've got this map, we still have to figure out where all these genes are, what are they doing, when are they becoming active, and when are they shutting off. It's a very complex process. But once we have markers, once we discover these markers that identify certain traits, then those are tools that breeders can use to, like I say, do a quick genetic test and know that they've got a particular trait. So let's think about how a breeder has conventionally operated over the years. And we've had some very successful peanut breeders that have come up with new, modern, and much improved peanut varieties. But what they do is they'll take two peanut varieties that have different traits. Like in this example, in the circles there, you've got a parent peanut plant that has good yield. We know that one yields really well, but it may be susceptible to a particular disease. So we've got another parent that has really good disease resistance, but it may not yield so great. So we want to put both of those traits together. So a conventional breeder would take those two parents, cross them in a greenhouse, and then you're going to get offspring from that particular combination of parents, represented by all the different peanut symbols there. Well, the first generation of a cross is very diverse. It's like those of you that have multiple children, unless they're identical twins, they're very different. You're not identical to your brothers and sisters. So just because your parents are the same doesn't mean you're the same. So this is the same that a breeder has to go through. He makes the cross, but then among all that diversity that comes from that cross, he's got to go find that one particular peanut that has those particular traits that he's looking for. So he'll take those lines, those different varieties, and he'll plant those peanuts out And it may be years before he knows that, hey, this one is yielding very well consistently, and this one has very good disease resistance. So it may take many, many years before that one line is discovered that he wants to pursue. With marker-assisted selection, you'll start with the same two parents. You're going to get the same diversity, but if you have that genetic test where you can go look for those markers, you can do the genetic test and know immediately this one's going to yield well and this one's going to have good disease resistance. And you can select that one line that you're going to pursue and start to grow out. So that's the advantage of marker-assisted selection, basically. It allows us to look for those traits, to get those traits incorporated into our new varieties very quickly compared to conventional breeding. The goal, of course, is an improved toolbox of technology for peanut breeders. That's what we're trying to do. We're trying to help the peanut breeder to make better peanuts. And we are trying to do that without the controversial GMO technology. GMO is a wonderful technology that has allowed other crops to make some significant improvements. We, as scientists, are not necessarily opposed to that technology, but we know the reality of the desires of consumers. So again, we're making this conscious decision to not go with GMO technology, and we feel like with this marker-assisted selection, we can accomplish some of the very same things and get some of these improved traits into peanut without having that GMO technology. And here you see an example of a statement on a product that's showing the consumer that it does not contain GMOs. Our Peanut Foundation has a website. If you go to the American Peanut Council website, there's a link there to the Peanut Foundation. And at that site, you'll be able to find all the technical reports that we've published over the last few years. There is a final report now on that website. Most of the report is very technical, and most of us would not understand a lot of that's in there, but there is an executive summary that's written in layman's terms that kind of summarizes better than I can in the time allotted this morning what we've done with this project. So we feel very good about this, that we're on the right track, and that amazing new developments are going to come about because of this investment in genetic exploration. So with that, I'm going to yield the rest of my time to one of the scientists that has actually led this work over the last 5 years. Dr. Scott Jackson, his bio is in the program, and I won't repeat that, but I will say that Dr. Jackson is one of the leading agricultural geneticists in the world. He is a very outstanding scientist with a lot of accomplishments, and we're very fortunate to have Dr. Jackson and his team working on peanut. So Dr. Jackson is not only an outstanding scientist, but he has a unique ability among outstanding scientists to be able to communicate to a bunch of non-scientists. So he can actually communicate, and you're going to understand what he says. So Dr. Jackson, glad to have you here, and I'll yield the rest of my time to you.
Which one advances? Good morning, everyone. So I'm from Athens, Georgia, which is only about 250 years old versus the centuries found here. So it's been nice to visit the other Athens, or the real Athens. And thanks to Darlene and Steve for the introduction. A lot of it segues into what I'm going to talk about, which is a bit more technical, but I'm going to try and keep it as simple as possible. And I have a tendency to start going faster and faster and faster as I talk. So someone please wave at me if I go, Patrick, wave at me if I start talking too fast, or throw something. So Steve introduced the peanut, we call the Peanut Genome Initiative, and sort of a background behind it and what it means. I'm going to go through a little bit more detail and sort of explain what happened and how we're planning to use it. I do want to reiterate one thing that Steve mentioned, and that this was an international effort. It was begun by the U.S. peanut industry, but it quickly gathered a lot of international partners from almost every continent, except for Antarctica, that it all contributed in very valuable ways to this initiative. So it's an incredibly, and it brought, I think one of the benefits of this project is it brought a lot of people internationally together, working together, that hadn't been working together previously. And that's been a benefit of this large-scale project. So Steve mentioned or introduced a little bit about what genome projects are. So everyone knows that, or everyone has heard about the Human Genome Project, which began back in the late 1980s. And the goal, sort of like a space project back in the 1960s, 1970s, was to transform the way we do biology, human biology. And the way to do that was to understand the blueprint or the genetic material inside of humans. And so DNA, what we're talking about is sequencing DNA. So that's the material in your cells that contains the genes. And DNA is simply just 4 letters, A, C, T, and G. But there's a lot of them. So you can see here you have chromosomes. The chromosomes are made of this strand of DNA, and you can see the A, C, T's, and G's in this DNA. And that's what we mean by sequencing a genome. We want to know all those A, C, T's, and G's that make up that genome, and then we want to superimpose on top of that what the genes are and how they control the traits. So you can see the Human Genome Project took about 13 years. It cost almost $3 billion. And about 3 billion A, C, T's, and G's. That's a lot of 4 letters. So like the space project back in the 60s and 70s, it spun off a lot of industries because a lot of technological advancement that happened during the development of the rockets and the computers and other materials. And the same thing happened during the Human Genome Project. When they first began, the sequencing was very expensive. That's why it had a price tag of almost $3 billion US. But technology advanced as they went along the project, and it got to the point that we could sequence the peanut genome for about $6 million in much less than 13 years. We actually did it in just a few years. And that's what we're showing here is just the white line is Moore's law, which is, the processor speeds double every however months it is. And the green line showing is the cost of sequencing. And so actually we beat Moore's law in the cost of sequencing. And that's what allowed us to do things like peanut that didn't have $3 billion. That's the entire industry for I don't know how many years. But $6 million is a much more manageable piece of research money. So this sort of reiterates some of the things that Steve said, but basically the genome is just all the inheritable traits of an organism. So the human genome has about 50,000, or about 25,000 genes that make up, that's what leads to what we are. So those genes interact, they produce proteins, and they interact with the environment to produce what we are. And you can think of it as a blueprint. And so when we have the genetic material, we have all these genes, we have a blueprint of what that organism, what that thing should look like. So you can have a blueprint of a 747. It doesn't give you the 747, but it lets you understand how the pieces go together to make that 747. And then you can think about engineering pieces of that 747 by breeding, like Steve talked about, to have faster engines or whatever you want. And in the case of a peanut, to be more disease resistant, more heat resistant, more drought tolerant, different composition, different flavor traits. Once we understand the genes that make up these traits, we can begin to engineer these using the technologies that Steve talked about, and that's putting DNA markers associated with those traits into the hands of breeders so they can produce lines that have those traits that they want, and much more efficiently and faster than they currently do. So basically, it's a framework that will accelerate gene to trait association so that we can take this gene or this set of genes controls these particular traits. We can begin engineering pathways, and not by GMO, but by engineering by breeding, selecting different lines that have different versions of genes, and advanced breeding generally. So a little bit about peanut. It's native to South America, and probably many of you know this. It was domesticated in Peru. Steve talked about domestication in dogs and peanut. That happened in Peru about 10,000 years ago. Darlene talked about its important protein oil source and all the other phytochemicals that come from peanut. Obviously, this group trades and markets peanuts around the globe, but it's also a subsistence crop. So there's many growers, farmers in parts of South America and Africa that rely on peanut as a food crop. And as I mentioned before, it has almost about 2.7 billion A, C, T's, and G's. So very close to human. I don't know if that makes you feel good or bad, that peanut has as much DNA as we have. Actually, peanut has more genes than we have. Maybe that makes you feel less important than what we should be. And Steve talked about the wild relatives, and that's something that we're really focusing on now, is now that we have the genome sequence, we have the genes, and beginning to understand what genes control which traits, we can go back to these wild relatives where we've left genes behind during domestication and bring those genes forward into breeding programs for traits that we're interested in. So how big is the peanut genome? I already told you it's about 3 billion bases. The human, about the similar size to the human. Human costs about $3 billion. Maize, corn, was about 2 billion bases, and that was sequenced several years ago, and that cost about $100 million. Peanut, larger than maize, a little bit less than human, it costs about $6 million. So if you think about a Bible has about 3 million letters. So really, the peanut genome is equivalent to about 8 or 9 Bibles. That many A, C, T's, and G's. So it's a good thing we have computers, otherwise we'd have a tall stack of paper looking at all these A, C, T's, and G's and try and figure out what they do. But just to give you a concept of how big the genome is, that's what it's equivalent to. All right, now I'm going to get technical for just a minute. I'm going to try and keep it so you don't fall asleep. It's early in the morning. For some of us, it's about 3 or 4 in the morning. I'm going to talk about some complicated genetics for a minute, okay? So you can either leave or you can try and stay awake. So I'm going to introduce a term called polyploidy. I'm going to explain this a little bit because this is something that complicates doing genetics and sequencing in peanut. So we humans are what we call diploid. And what that means, diploid just means 2. It means you have 1 chromosome from mom, 1 chromosome from dad. And that's what we're showing in red and blue here. So you have chromosome pairs, and each set of chromosomes, you have 1 copy from mom, 1 copy from dad. That means for every gene, you have 2 copies. You have a copy from mom and a copy from dad. And so we can think about traits. The inheritance is, if the trait is dominant, if you think back to Mendel, the flower colors, there's dominant flower colors and non-dominant. We have similar traits in human. So you get chromosomes from mom and dad, and here's a simple trait that's controlled by a single gene in humans. And so your earlobe, detached or attached, the detached is dominant. And that means 3/4 of the population has a detached earlobe. And 1/4 of us have an attached earlobe. Okay? That's how diploid genetics work. If humans were polyploid like peanut is, that means you'd have 2 copies from mom and 2 copies from dad. And instead of 2 copies of the gene in each cell, you have 4 copies. And that just makes the genetics much more complicated. So if we take the same trait, that means the ratio would be only 6% of the people that have attached earlobes. So if you think about this in the breeding context, and Steve put up that beautiful illustration, you take a variety A times variety B, and you have this population, that means if there's a trait that you're looking for that is recessive, like the attached earlobe, fewer plants in that population will have that trait. And it makes breeding that much more complicated. So if you are selecting for a trait, and you have a diploid genetics like humans, you make a population of 100, and 25 of your plants will have that trait you're looking for. But when you have these polyploid genetics, you have to make much larger populations to find those traits, and that just makes things more complicated. It also makes sequencing more complicated. So sequencing the genome, instead of a genome in the nucleus, we now have 2 genomes. So that was one of the things that complicated peanut. So instead of, in human, we're diploid, we have 1 genome, but in peanut you have 2 genomes. And so in the process of sequencing peanut, we're sequencing 2 separate genomes in the same nucleus. And I'll explain that just a little bit more. So Steve put up the slide with what the wild peanuts look like. And the approach that we knew that 2 different species came together about 10,000 years ago to make the modern peanut. And each of those species donated 2 chromosomes, and that's why we have polyploid genetics. And so the approach we took was we have representatives of those species in the collections around the world. And so we took 2 representatives and sequenced those, which is basically like sequencing a human, they're both diploid, to understand what those parents look like. And so this is a diagram what it looks like. So now instead of 1 copy from mom, 1 copy from dad, you have 2 copies from each of these wild species. So we sequenced those two diploid ancient species, and this was what Steve referred to as published 2 years ago. And that gave us information so that we could then think about sequencing the cultivated tetraploid, polyploid peanut. So if I put this in another way of sort of illustrating this. So when we sequence a genome, you have these 3 billion A, C, Ts, and Gs. We can't sequence each chromosome from beginning to end with the technology we have. So what we do is we cut it up into random fragments, we sequence those fragments, then we use computers to put it all back together. So it's like a giant jigsaw puzzle, but instead of 500 pieces, you have 3 billion pieces. So it's much more complicated. So you can think about taking a picture, make a jigsaw puzzle, put it back together. That's relatively simple. In the case of peanut, we have two somewhat similar pictures in the same genome. You have one from the one parent and one from the other parent. They diverged a little bit over time, so there's some differences between them, but there's a lot of similarity. So if we break them up into billions and billions of pieces, it's very difficult to make sure that this piece goes here as opposed to over here, because there's so much similarity between the two. And that's why we sequenced the two ancestors, those things that donated the chromosomes from the ancestors, separately before we did the polyploid peanut. So each of those is about 1.3, 1.4 billion pieces versus 3 billion pieces, and each one has about 25 to 30,000 genes. So we sequenced those, and then we were able to sequence cultivated peanut down here. So in actuality, that $6 million, $3 billion to sequence one human genome. With $6 million, we sequenced 3 genomes, the 2 ancestors and the cultivated species that we grow in the field today. And we actually sequenced a variety called tip runner. So really, that's just the beginning, and I think Steve touched on this. The goal is to put tools into the breeder's hands or the physiologist's hands or the people working on how you grow these plants in the field, or the understanding the traits that you as marketers want, or you as the people putting them into candy bars or other products want. So making sure they have the right oil concentrate ratios, making sure that they have the right flavor profiles, storage profiles, anything you can think of that's controlled genetically. We can now begin to drill down to the genes and control those traits, put tools into the breeder's hands so they can begin to mix those traits together into the varieties and that the growers grow. So this was done as an international collaboration. All this data, if you really want to go look at it, you can go look at it now. It's a website called PeanutBase. And this is what it looks like. This is a segment of the genome. These are genes down here that have been placed upon the genome. And what this is showing the expression pattern where these genes are expressed in the peanut plant. So just as an example, this gene here, so what we have here is leaf, nodules, different parts of the plant, and these are seed, parts of the seed. And you can see that this particular gene is expressed only in the seed. This gene down here is expressed in all sorts of different tissues. And so we can use this information, like Steve talked about, to begin to understand what are the genes that control seed development, control the different oil, protein compositions, how they're expressed, and begin to mix and match those together to engineer plants without using genetic modification, but using standard breeding processes. So a number of years ago, there was a study showing suggesting what are going to be very disruptive technologies in the next 25 years, up to 2025. These are things that would be disruptive in the trillions of dollars. This is from a McKinsey report. And it includes things like mobile internet, Internet of Things, cloud, robotics. And number 7 is next generation genomics. And they estimate this to have an approximately $2 trillion impact over the next 20 to 25 years. Obviously, most of this is in human health and medicine. So with the human genome, they're now developing ways where you can have personalized medicine. So based on the genes that you have and how they respond to the environment and different medicines, they can personalize the way that they develop treatments for whatever illnesses you might have. But part of that is agriculture. So the application of genomics and breeding means that we get much better varieties much faster, much more quickly than we currently do. Impact on how we package traits into those varieties. So I think it's going to really have an impact in peanut in the future. It's had an impact in other crops, and we're now beginning to see how this is working in peanut. So Rob Fraley, who's the vice president and CSO of Monsanto Corporation, was once stated that genomics reduced their time to variety development by 50%. So If you think of this in the context of peanut's about 12 to 15 years for variety development. So if we really apply genomics, we should have that down to 6 to 7 years for new variety development. And that's the goal, is to make sure that the breeders and people developing new lines and that with the traits that are wanted by the community are able to get those out much more quickly. Already, markers have been used in peanut breeding, so just being able to track specific traits. So these are what we call DNA markers, so they can track specific genes that control specific traits and put those into varieties like Steve talked about. And so there's when the push to bring out high oleic peanut, a marker is developed to track high oleic trait, and then they were able to introduce that trait into existing varieties much more quickly. Steve touched on this. This is one of the foci of what a lot of us are working on now, is now that we understand the genetic makeup of cultivated peanut and its ancestors, we're trying to go back and pull genes out of the ancestors using traditional crossing and breeding to bring those traits into cultivated peanut. And a lot of those are disease resistance, pest resistance, nematode resistance, things that don't exist in cultivated peanut because of these domestication bottlenecks that Steve talked about. And we're really beginning to understand the genetic control of complex traits. And this will take a lot of work. So, for instance, flavor is a very complicated thing. It's complicated to measure. And there's a lot of things of genetic, environmental and genetic things that lead it to a flavor profile of a peanut. And so it's going to take a while to get there, but I think we're beginning to understand we'll soon have a better understanding how these things work together. So I want to finish with this just to show where we're headed. So we've sequenced these two ancestors of peanut, and we sequenced the peanut that is grown in the field and that is marketed around the world. And Steve mentioned about this domestication. So, in the case of dogs, the dogs that you have in your house do not represent the dogs that are in the wild or what they came from. And it's the same thing with plants. Plants are domesticated from things that don't look very much like the plant that farmers grow. So if you think about maize, for instance, the wild maize plants, the seeds fall on the ground. So if you're a grower, you don't want that. You'd have to vacuum up all the seeds off the ground instead of just threshing the plants. And it's the same thing with peanuts. So seed size, the way the seeds come together within the pod is much different than in the wild. And this is done through what we call genetic bottlenecks. So domestication is a genetic bottleneck. So that means you leave a lot of genetic diversity behind. A lot of bad traits are left behind, and you bring forward good traits so that the seeds don't fall to the ground. You have larger seeds, and that things flower at the same time, they mature at the same time. And then you have the bottleneck of modern breeding, which is further reduces the genetic variation. You get rid of the things that you don't want, and you increase those that you do want. So diagrammatically, so we have these ancestors, what we call the A and B ancestors of peanut. And about 9,000 to 10,000 years ago, they came together and formed a polyploid peanut. And then there's domestication and breeding. And so the genetic variation gets narrower, narrower, and narrower, and narrower. So if you think about this, maybe represents the genetic variation in the A species, the genetic variation in the B species. Only one of each one was selected. So there's a lot of potential genetic variation that's left behind. You can think about it like your mom and dad. So, since I'm a male, I'll use it. I selected a wife out of the 3 to 4 billion candidates on the planet. So that I selected one out of 3 to 4 billion. That's a lot of genetic variance that I ignored. And it's the same thing in peanut. There's a lot of genetic variance that we lost just because these two representatives of the A and B came together 9,000 to 10,000 years ago, and then we did breeding and selection, domestication following that. And so what we're doing is we're going back, exploring the genetic variation. A lot of it's stuff you don't want, but there's a lot of good stuff that we do want. You can build more resilient plants to climate, to heat, to stress, to disease, because those traits are back there. And so what we're doing is we're selecting accessions or different representatives of these different gene pools, what we call A and B gene pool, recreating the polyploid peanut, and then beginning to cross genes using traditional crossing methods into cultivated peanut. And I think this has already happened decades ago, but it was a very laborious process. We're doing this in a matter of years now. And I think it's very exciting because there's a lot of interesting material that's coming out of that. And it's done using traditional methods, not genetic engineering, which I think is important for the broader community. So we have lots of accessions, these maintained around the world at different places. Within the US, we have about 10,000 accessions of peanut and its wild relatives. And we're exploring these using modern tools that are all premised off of this genome sequence.